So we have already seen about the Mendel's law of inheritance, that is the three laws, that is law of dominance, segregation, and independent assortment. Okay, so in law of dominant, we already know dominant means the character which will show the dominance will get expressed, other one will be a recessive one. Then law of segregation. Law of segregation means what? Each pair of genes, if you take, they will separate during the gamete formation, so that each gamete is going to carry just one particular trait or character. Whereas law of independent assortment means what? Whatever alleles were there, that are separate for the contrasting traits. And when there is a, what do you say, a division, then each of them are passed independently during the gamete formation. Out of this three, we have two types of crosses, that is monohybrid and dihybrid. So the independent assortment is there, that is for the dihybrid cross. Okay, so today we are going to see how to draw Punnett square and how to put the characters and get the F1 and F2 generation. Okay, so basically, as I already told you, that Mendel has taken 7 contrasting characters. Seven contrasting characters of the pea plant. Okay, so first one, the color, position of the flower, then the seed color, then the shape of seed, then pod shape as well as pod color and the height of plant. So these are the 7 contrasting characters, and out of that he took one as a dominant one and the other as a recessive. Okay, so whatever dominant characteristics are there, that was purple color flower, recessive was white. In the position, axial and terminal. Seed color, yellow and green. Seed shape, round and wrinkled. Pod shape, inflated and constricted. Pod color, again green and yellow. So if you see pod color and seed color is the opposite, showing the dominant and recessive character. And the height is, we already know, tall and dwarf. Okay, so taking any of the two combinations, we are going to see the monohybrid cross and the dihybrid cross. So the first, let's move on towards the monohybrid cross. Monohybrid cross. Okay. So monohybrid cross means we already know that here only one, gene and one character is involved. One gene, one character. Okay, so here the examples will be like AA or BB. Okay, it may be either both recessive, then one recessive and one dominant, or both dominant, anyhow. Okay, and whatever Punnett square you are going to draw is 2 into 2. Will be the Punnett square thing. So, like easily if you want to see towards it, and if we take the characteristic as a tall and a dwarf, how will a monohybrid cross come? So the first thing is, I'll just show you the direction. So here we are going to take tall and dwarf. So the example will be one homozygous, tall, homozygous. So similar it will be. So the genes for this homo means what? Capital TT will come. Whereas into one heterozygous. So heterozygous tall means what? One capital T and one small t. Fine. So whenever you're going to draw a Punnett square, so now we have the TT into TT, right? So here on this side, upper side and the lower side. So in the upper side, the gamma is from the first parent, whereas over here, it will be from the second parent. So here you have to write it as capital TT, capital T, small t. Okay, so everything will be same. I'm just going to show you how you have to write this down. So if you take the first scenario, okay, so over here, in the first thing, what you have to do is you have to merge the first capital T and second capital T. So it becomes T, T. Okay, so this is the first how you have to do. Then the second one, which is there, in this, what you have to do, you have to take a T from here and you have to take a T from here. So already TT we know, here also capital T, capital T. This is the second step how you have to do. Third step is we'll take the lower ones. So here, this capital T and this small t. So capital T, small t. So over here already it's there. We'll just write it down. And for the fourth, it will be from here and from here. So again, capital T, small t. So yes. So this is how you have to combine the gametes of the first parent with the second parent to get the products. So whatever 4 you have got 2 by 2 square of the Punnett. Okay, but the same thing when you go to a dihybrid cross. Here what happens is instead of one gene, one characteristics, here you will be having 2 organisms. But they will be what? Heterozygous. For getting what? 2 separate features. So thereby here obviously 2 organisms and heterozygous it is. So obviously the Punnett square will be big. So it will be 4 into 4 thing. Okay, so here now we are going to see properly how the F1 and F2 generations are there. Okay, so first let's see the F1 and F1 generation of the monohybrid cross. Now we already know how to do a Punnett square. Okay, so let's go in detail. Okay, yeah. So I'll just write it down. First is the parent generation. See in this topic, in this chapter, this topic is very important. You need to know how to draw this. So tall and dwarf. This is how you have to write it. Okay, the gametes you're going to take. Here it's a capital TT as we already know. Dwarf means small tt. So I'm going to take one T from here. Then one T from here. And I'm going to get TT. So whatever I have got over here is tall plants. All plants will be tall over here. Okay, because if you see over here, capital T is there everywhere. So all I am getting is what? Tall. Okay, so this particular thing which I have got is the F1 generation. F stands for filial. Okay, so first filial generation this is. Now, as per the Mendel's law, what we are going to do? We are going to self-pollinate the plants. So self-pollinate means F1 into F1. So self-pollination. So that will be F1 is this one. So it will become TT into TT, which is the product of F1. And now we are going to form gametes for the same. Okay, so the gametes over here, we'll be getting 2. Okay, we'll just separate it out. And after separation, now we want to get the F2 generation. Okay, so for getting the F2 generation, what we'll do, we'll just cross this out. So I'll just write it over here. capital T, small t, another capital T and small t. So now when you're crossing this, how it will come? First thing is you have to take this first T from here and then you have to cross with this, the first one. See, instead of the Punnett square, I'm just doing it over here in this way. Okay, so it will come like this. In the same way, again, what you have to do, the first T, so you're getting like this. Now the first small t and the big T. So it's a capital TT. And again over here, a small t and a small t. So you're going to get over here is what? TT. So here, this one will be your F2 after combining this both. So here, if you see, capital TT means tall. One capital T, one small t means again tall. Again the same. Whereas over here, if you see, two are small, so it will be short or dwarf. So this is your second filial generation. So here, if we see the phenotypic ratio and genotypic ratio, it's easy to determine because phenotypic ratio is accordingly, we see 3 talls and 1 short, so it's 3:1. Okay, whereas whatever the genotypic ratio is there, it will be 1:2:1. Why? Because if you see over here, okay, this is 2, this is 1, and this is 1. Okay, so this is 1:2:1. So this is how the characteristics will come over here. So either you can take the homozygous or a pure condition where either both will be dominant or both will be recessive, or you can take a heterozygous condition where one will be dominant and one will be recessive. Okay, so whenever you're crossing in the monohybrid cross, this is what you will get. Okay, so now in the same way, I hope this is the like the monohybrid cross is clear. So now in the same way, we are going to see the dihybrid cross, but in the dihybrid cross, there will be two heterozygous characteristics. So let's see accordingly. So I'll just write it down. First, again, the same pattern we are going to follow. Okay, so here the parent. So here we'll take round and green seeds, okay, plus a wrinkled and yellow seeds. So then the gametes will be, now instead of 2, here 4 alphabets will be writing. So capital R, capital R, capital Y, and wrinkled yellow is recessive, both. So we'll write small. So in the F1 generation, whatever you're going to get is one capital R and one capital Y, and here one small r and small y. So when you merge together, you're going to get capital R, capital Y, small y. This is nothing but a round yellow seed. So round yellow seed will be the product of F1 generation. Now again, as we did in the mono hybrid cross, that is the self F1 pollination. So here also we are going to do the same thing. So selfing the F1. So it'll be like again F1 into F1. So we already got the products out. Capital R, capital Y, small y. So now we have to just, distribute it in, because we have to do a Punnett square. So we are just going to distribute this thing. So if I take one product of this, because it's 4/4, so 4 things I'm going to write over here. So one capital R, one capital Y, one capital R, then we'll take one small y, small r, then capital Y, and small r, small y. Okay, so because it's F1 and F1, this thing you have to write 2 times. Okay, and this thing again, you are going to get 2 things like this. Fine. So this will be the gametes altogether. Now, whatever 4 characteristics we have got, or the 4 distribution we have done, let's put this into a Punnett square. So I'll just draw a Punnett square. So whatever characters we have distributed, let's just fill this ry. Then this ry, small r, capital Y, then small r, small y. Same thing, write it on this side also. Mhm. Okay, fine. So now let's just cross this. So here it will be r y y r capital R, small y. Yes. Okay. R. Y y. Okay. So this is how we are going to get the dihybrid cross Punnett square. Now, if I see the phenotypic ratio, it will be quite easy over here because just we have to see which one is round yellow, round green, wrinkled yellow, and wrinkled green. Okay, so how it will be? So I'll just write it over here. According to the characteristics that we have taken, round yellow, then round green, wrinkled yellow, wrinkled green. Okay, so we have to count one by one. If you see the first one, Okay, now round and yellow was the dominant character. Wrinkled and green was the recessive character. So accordingly here, two dominants are there. So you will count it as one round yellow. Okay, so like this, you have to count all these boxes. So ultimately, whatever ratio you're going to get is 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. Okay, 1 wrinkled green is over like here, the small one. Okay, so 1 wrinkled green. So accordingly, if you take the genotypic ratio over here, it will be a very big genotypic ratio because you have to write all this and you have to calculate it. But if you want to take a genotypic ratio of this particular thing, it will come as 1 is to 2 is to 2 is to 1 is to 4 is to 2 is to 1 is to 2 is to 1. So in monohybrid cross, genotypic, phenotypic both are easy to count. But when you're taking it into a dihybrid cross, because we are having two different organisms with two contrasting traits, hence it's hard to find out the genotypic ratio because you have to count all the thing and then put it into a ratio. That is the thing. Okay, so this is how you're going to do a dihybrid cross. Okay, so moving on towards the next part, which is the sex determination. Okay, so now over here, obviously two types are there. Okay, that is the male and the female. So with respect to that only, we are going to see the sex determination. Okay, so now here we'll be taking two parents only. Okay, that is father and mother. Okay, so whenever we determine the sex of a newborn, it will vary according to different species. Okay, but after all, whatever the fusing will be there, that will be same. Fine. So this all is dependent upon what the chromosomal characteristics which we are having. Okay, so like if you see there are different, like basically two types of factors are there for which are responsible for the sex determination. Okay, that is one which is called as the environmental factors, and the second one which is called as genetic factors. Okay, so environmental factors means it's applicable to the animals because some animals they lay eggs. So for them, this is most probably applicable. What happens in the environmental factors is if you take some animals, they live in different habitats. So the temperature, okay, where they live in, it will be different. So because of that, the egg fertilization also, depending upon the habitat and the temperature, after the egg gets fertilized, what thing is going to come out, then only we'll know. Okay, so that will decide the gender. If it is what environmental factor decides the gender. For example, you can take turtle. Whereas if we take a genetic factor, it will be depending upon the DNA or the genetic factors which are there. So humans are the greatest example over here. Okay, so humans gender, that is individual, is determined by how? Chromosomes, pair of chromosomes. And we have 23 chromosomes, out of which 22 and the 23rd one. Okay, so whatever the 23rd chromosome is there, and this 22 is there. 22 chromosomes are called as autosomes, autosomal chromosome. 23rd pair, which is also called as the gamete chromosome or sex chromosome. This is called as allosome. Okay, so the first 22 chromosomes, which are somatic in nature or vegetative, you can say, they are what? Autosomes. Whereas the last gametic chromosome, which is there, it's called as what? Allosome. Okay, so whatever this 23rd chromosome is there, that is going to determine that the going to be born baby is of which gender. Fine. So if you see over here, individually, if I take a female and the male, Here it will be X, 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 Y. So if you see, for the female, the chromosome is homo, whereas for the male, it's hetero. So depending upon which X or Y from the male is going to join to one X of the female, depending upon that, the gender of the baby will be decided. Means the gender of the baby is decided by whom? The gamete or the chromosome from the father. Okay, so if we take like this, in the same way, how we took the monohybrid and dihybrid cross, we take the gametes over here, that is XX and XY. Okay, so separately, we'll write it over here. And then when you combine these things, you're going to get a zygote. So in this, the zygote will be like, there will be one XX, then there will be again XY, XY. Okay, so probability will be 50% over each when this is combined. Okay, so 50% of a female child and 50% of a male child. That will be the probability when there is a fusion of a XY and the XX. Okay, so this is how the, the sex determination is being done.